The alpha-specific genes include, as I indicated before, the pheromone gene. So there are actually two alpha pheromones, and there is a receptor, and there are also genes for processing the pheromone. The pheromone is synthesized as a pre-pro pheromone, so it has to be part of that original polypeptide have to be removed to have the active pheromone. All of these are alpha-specific genes. They are required for mating as an alpha cell. So if you have mutations in any of these genes, this cell cannot make as alpha. Now, the A-specific genes are off because alpha-2 represses them. This block arrow, that's what it indicates, is repressing this gene. And haploid-specific genes are expressed. They're on, but they are not regulated by alpha-1 or alpha-2. That's another mechanism. I'll come to this later. One example of haploid-specific genes is RME, one which is very important, and I'll come to this one in A alpha cells. Okay, now look at the A allele. Mat-A has one gene, and it's A1. Okay. A1 codes for a homeodomain protein. Remember that alpha-2 codes for a homeodomain protein. This one codes for a homeodomain protein, but these are two different classes of homeodomain proteins. Homeodomain proteins are classified into two major groups. A1 belongs to type 1, alpha belongs to type 2. That's, the details of that are not important for understanding. So in Mat-A, you have the RNA polymerase binds to the motor. Remember, this is the operator site. I forgot to label this operator promoter. But there's no alpha-2 here, so the RNA polymerase binds and transcribes A-specific genes which reside throughout the genome, but not at the MAT locus. So these genes are on because there's no alpha-2 repressor. Therefore, nothing is preventing RNA polymerase from binding to the promoter. Okay, again, so the Mat-A allele contains one gene, Mat-A, and it also encodes a homeodomain protein. Okay, so the Mat-A gene, so you say there is a homeodomain protein, and here, A1 is not doing anything here. It's not activating any of these genes. This protein is present, is not required in this cell. Okay? Only RNA polymerase. These cells are expressing these genes because there's no alpha-2, not because you have A1. Okay, and Mat-A1 is not required to express any specific genes. And how do we know this? Well, if you delete allele, you delete Mat, the cells behave as A, and they will produce the A pheromone and the receptor for the alpha pheromone. That's the default state for yeast. So if you have an alpha cell, and now you go and delete that the mating type locus, in other words, the Mat alpha allele, now the cell is going to behave not as an alpha, but it's going to behave as an A. Anytime you delete the mating type locus, you have an A cell. This gene is only required in the alpha cells. So let's summarize here. So A1, A cells have the Mat A allele the Mat-A allele has one gene that codes for a homeodomain protein. Alpha-specific genes are not expressed in this cell because there is no alpha-1. There is no alpha-1. You require the activator alpha-1 to express these genes. So these genes are off. Remember, these cells have both type alpha-specific and A-specific genes. So these are off because you don't have alpha-1. These A-specific genes are expressed because there is no alpha-2. You can delete these genes and still you express A-specific genes. And the A-specific genes include the A pheromone genes. Again, there are two A pheromones. And there is one gene for the receptor for the alpha pheromone. And you require also genes for processing the A pheromone, which is secreted via a non-conventional secretory pathway. So you need certain genes to do that. These genes are required for mating as A. If you don't have them, this cell doesn't mate as A, no matter what you have the mating type locus. Okay, and haploid-specific genes are expressed, so they are on, and again, RME and next. Okay, so that explains this. So A cells, why they have they express A-specific genes but not alpha-specific genes. Alpha cells, why they do not express A-specific genes but express alpha-specific genes. And now we have A-alpha cells. Okay. In A-alpha cells, there are two MAT alleles, A and alpha. So why doesn't it express this? Why doesn't this cell type express the pheromone genes or the receptor genes? Why doesn't it express haploid-specific genes? Remember, haploid-specific genes are expressed in both A and alpha, but not here in the haploids, yeah. So, and why does it undergo meiosis? Okay, so in the A-alpha diploid cell, you have alpha-1, alpha-2, and A1. Here in this cell, you are going to be making these three proteins. A1 is going to interact with alpha-2 to form a new protein, a heterodimer consisting of two different homeodomain polypeptides, A1 and alpha-2. This is a heterodimer, and it is a repressor. Okay, this is a repressor. So, this repressor is going to bind to the operator site, which is composite. This is A1 alpha-2 operator site. So it has two sites. To one of them, you bind the one moiet, and to the other one, you bind the alpha-2 part. And this represses expression of haploid-specific genes. This is why haploid-specific genes are off in A-alpha diploid cells. You do not have this heterodimer in A or alpha cells. You cannot repress these genes. Now, this heterodimer also represses alpha-1, this gene here. There is a dual operator site that has an A1 and alpha-2 binding site. So now you have repression of alpha-1. If you have repression of alpha-1, that means that you do not express alpha-specific genes, but you require alpha-1 to express alpha-specific genes. You do not express A-specific genes because you have also alpha-2 by itself that is going to be resisting the A-specific genes. So, in a big summary here, so you have alpha-1, alpha-2, this is part of the MAT alpha allele, and A1 to the MAT-A allele. So these reside in one cell. And you have that the alpha-2 and the A1 form a new combinatorial protein. Combinatorial because you have the action of two different proteins coming together now and performing a function, a new function. Oops. So this consists of, as I said, of two different homeodomain polypeptides, and these are carries functions that are different from those of ALF2. So alpha-specific genes are not expressed because there is no alpha-1. And why is there no alpha-1? Because this new combinatorial activity, the A1 alpha-2 head dimer, represses alpha-1. So you do not have the activator. Alpha-2 represses the A-specific genes. These genes are off. So this is why you do not express for pheromone, the pheromone gene or the receptor genes for alpha A. So you do not express the A pheromone genes, you do not express the alpha pheromone genes, you do not express the A receptor gene or the alpha pheromone receptor gene. They are all off. And the new combinatorial activity, A1 alpha 2, represses haploid-specific genes. So RME is a haploid-specific gene, and so it is not expressed. Okay, and I'll tell you this is a very important gene that I will tell you in a moment about this function. So this is the reason all these genes, alpha-specific, A-specific, and haploid-specific genes are all in the A-alpha diploid. So why does it undergo meiosis? So this is just recapping in words what I just said. So A-alpha cell, the A-alpha cannot mate with A or alpha because it does not produce the pheromones or the receptors for the respective pheromones. It can undergo meiosis because it represses RME, which is a repressor of meiosis. Haploid or A or alpha cells cannot undergo meiosis because they express the repressor of meiosis. Okay, so this is summarized here. So in the A-alpha diploid, you make this new combinatorial activity, the A1 alpha 2 repressor, which represses RME. If you have this repressor, RME, you are able to repress IME, and IME is an activator of meiosis. Okay, so let's go and do that a little bit of a slow motion. So here again is that relationship, and what happens? So in